Welcome to everyone to the breakout session. We will be today discussing spatial technologies. So I'm Ana Pascual, and I'm part of the task force on spatial genomics, together with Stefania and Aruta, led by Holger. And I'm also leading the spatial genomics team at CNAP, the National Center for Genomic Analysis in Barcelona, Spain. Also, maybe not a leading country.
You're on the video.
All right, so welcome. We are here basically today to discuss about spatial technologies and to discuss which of those are actually suitable for Atlas production, and to see how can we drive their implementation in the next phase of Atlas building efforts. And for that, we have to consider several aspects of the different spatial texts out there. We've written down here some of them, but we are basically here to also hear from you your thoughts and your experiences, right? So not all the technologies are available everywhere. That's one point that we have to take into account if we want to build Atlas across regions and countries. Also the readiness for the different technologies for large-scale cell Atlas generation, instrument robustness, the data types that we get out of the instruments, spatial resolution, plexity, sensitivity, specificity, scanning area, turnaround time, etc. So now we'll just put some questions that we thought would be interesting to discuss with you, and we sort of ask you to contribute to an open and dynamic discussion. So first one, what technologies and importantly, what modalities should our community consider to build a spatially resolved human cell atlas? And within that frame, let's say, are there redundant complementary technology pairs, not only within modalities, but also across modalities? Thoughts?
So before deepen, okay, I don't have to use the microphone here from here. So before deepening the discussion, I'm just curious how many people here in the room have a direct access to any of the platforms like the Zenium and the Visium and the Cosmics and whatsoever. And maybe, okay, of course, you're responsible for all of the things. And so how many? Of course, you and then not so all people. And okay, so then my question is, so how then and how you are planning when we want to start the single cell spatial analysis? How we should do? For example, there is a way to ask. Say, define a commercial company, maybe sometimes outside of the country. So, and how do you think about? So, can I? Yeah, you. Yes. I'm sorry for the interruption because I heard from you that time is quite limited.
Go ahead.
Go ahead. Sometimes IRB is an issue, and IP intellectual property is an issue, intellectual property is an issue, intellectual property is an issue. And sometimes it's not easy to send out the samples across the country, the ports, right? So, and I just want to hear some opinions from the airports, for example. Yes. Don't be so casual. I'm not.
So yeah, brought up from China. Right? So I guess that quick question would be like to stand on a guidance shoulder. So for example, like collaborate with yourself, you have the access. But in our case, we were lucky that quite a few companies offer trials. So we have test quite a few technologies already, and I think with that, at least in Thailand, it's quite difficult to get the instrument, especially when it's expensive. So when we gather some data and we able to publish some of this premium data, we can use that as a leverage to ask the institute to get the machines.
Yeah, but obviously Thai is a big country, so naturally the people have already started to think about having your own instruments.
Right, yeah.
Are they thinking that's risky still?
I think it's always come with risk, and I think you're aware of this. yourself as well. I think there are a few things that we have to be concerned when we getting one.
Because that was the case in Japan too. Because I myself have to convince the governmental officials.
Yeah. So I think one concern is that it's going to be underused. So I think that because it's quite expensive technology, right? So if it's not going to be highly used, it's going to be concerned obviously. And I think the other one would be that in China at least, because I think it's Spatial, people see it as something that can be worked with FPE. So if you have all machines already, then all the machines would be. In Thailand, we still public universities, so all the machines supported by the government instruments. So if there's one already, then it's going to be difficult to get a second, because you can just go and use the one that happened before.
Can I collect some more opinions from other countries? Because Thai is some of the Asian country I know, right? So, and how about the people from other countries, like European countries or outside of, say, Europe, Asia, or where, for example, Africa or Americas, or in person from any parts of the world?
But I think that the idea is one step back, right? So
Yeah.
now imagine that we would have access across, right?
Mm-hmm.
I think that we have to first define which are the techniques that we find are most or better suited.
Maybe we have to have a discussion about what technology.
Exactly, and which model.
You're right. So please, I'm sorry for the.
So I'm assuming that HCA until now has basically built atlases based on gene expression.
Regardless of the country.
So RNA, I assume, is a must. But then, do we believe that beyond RNA, we also need another method to build atlases?
I think the basic cheap one is HCA, which gives us a lot of information and will help us translating information. So I think HCA is fundamental. And of course, the more we can do, the better. But if one has to say what's the minimum, in my opinion, is RNA and HCA.
Completely agree, especially to then interpret the gene expression and data on a more, let's say, pathology-oriented or clinically oriented way.
So
how about protein? I like that.
No. I must go not to say so.
Okay.
You're right that traditionally the others were big on RNA because the technology was much more advanced. On the protein side, I think many of us use protein, but utility in terms of atlas building was being on the immunophenotyping side. And even there, at least for us, we have used it a lot that go beyond what we've seen with RNA. We have a finite range of phenotyping based on protein. Was a bit more functional readout, yes, but there was not that much of an added value. I think that's changing now a lot. Spatial technologies and within the protein side is much more established within the spatial readout, and seen on very good examples this morning. So protein, I think, has to be on the main stage. How well it can be integrated with the RNA-based phenotyping approaches is something I think we have to see. But at least on the technology side, I think protein is where RNA, or for a long time where RNA is. TCR has been used a lot, especially for T and B cell activity profiling and specificity profiling. And as we've seen this morning, I think it's in the spatial context, it will give very high added value working in kind of immuno-oncology space, where we see go beyond that positioning with T cell specificity and even tracing.
Any comment from the protein side? Maybe you're more familiar with protein side.
I think just echoing what Olga said, for anything clinical without the protein, you're not going to get adoption from the pathologists. They look at protein all day. I think when we think of experiments, we run the same slide RNA and H&E. You get the H&E for free, and then serial section protein. But now you can see a lot of the companies doing RNA protein and potentially HME on a
On the
same
same
slide. So I think that is at a minimum, if you have those 3 layers of data, that's important, because if you're thinking about pathology and HME, you need some layering of protein information.
Microphone is fine.
As we have people here developing technologies, the problem that I see at the moment with proteins that we rely on antibodies. Antibodies are not always there, they're not always good. So if we have to think what we'll need moving on, can we develop more antibodies, better antibodies, or just technologies that are antibody dependent? That's just a comment to where I hope on importance.
Unfortunately, we are having a person from a company which has just started to generate the protein analysis, already started the protein analysis. So you will
I'm just curious when you start.
So how do you think of the protein analysis in addition to the messenger RNAs? For example, in general or the more precise way?
Yeah, I don't have magic answers on the antibody, unfortunately, but I just wanted to ask one clarifying question. What you just brought up, like, what are the main challenges you're seeing? Is it just the availability of content, or is it you have content and then when you actually go and run it, the performance is more questionable than you thought?
What happens is the antibodies are not. I think if you buy an antibody, your chances that will detect what you expect is 50%. That's my pure guess. Huh? So how many times we buy, so sometimes you don't find the antibody at all.
Yeah.
Sometimes the antibody will be commercially available, then you use it and it's really not working. So you might have nice signatures at the RNA level, but then you can't validate it. You can't add that protein level because simply there is no the tools available to do it. I think that is a big limitation because we're finding new markers, the gene level, but then we can't do it.
Got it. That's useful feedback. Yeah, I would say the vast majority of time we spent developing our protein products is actually trying to convince ourselves that the antibodies that someone says work in a broad set of contexts.
So any comments? Yes.
I mean, there's another Atlas project, which is a protein atlas. I think they've done what we are doing, what we're discussing now to extend over the last 10 years.
Yes.
So far we have been working in separation, but now being towards spatial and having the protein more importance on the roadmap, maybe with time.
So I'm just curious. If there is any plan to combine, integrate the results of the human cell atlas, the RNA aspect with the protein atlas in more spatial ways. Is there any plan?
Now,
You are starting.
it will make more sense.
Uh-huh.
Starting now working in the spatial context. So far, there hasn't, or there's very few directions.
That's an issue of discussion. Can I continue this discussion because that's important? And of course, the more if all the layers can be considered included in human cell atlas, it's very of course everybody agrees that. But the issue is my impression is my background is transcriptome. So the protein analysis, especially when it comes to the antibodies, is technically more difficult and may not be suited for the starting up countries in the developed countries. From an Asian country like Japan, we are also concerned feeling like some the Asian people are less represented in terms of the human genome sequence and the messenger RNA, the superficial coverage of the messenger RNAs. So maybe to catch up with the current progress of the human genome sequencing and the simple single cell profiling, that is the issue of the first priority. And that's me comment. There may be an opinion still, but I feel that we got it and you got it. So Reguant-san, we have insurance. I'm sorry for getting you involved in from the comments from Japanese institution. We can do some more. So yes, yes, sorry, I'm still here.
Yeah, so the The quick question, sorry.
Question is that Asian people, my feeling is I may be wrong, the Asian populations are less represented in terms of the international database like human cell atlas. So, and how we can we can catch up with things, or just even though the number may be smaller than those, but we can jump into those advanced. I can say advanced applications.
Yeah, thank you.
The choice is quite difficult.
Yeah, that's a great point. Maybe I think the type of donuts on top of these molecules. Yeah, but the molecules, I think RNA is quite important as you also discussed.
Generally, we are talking about this healthy ones.
Mm.
So the cataloging the gene expressions,
Yeah, like
and it
age
started with messenger RNA expressions.
Yeah, like age groups.
Yeah. So the field is already kind of mature in terms of the messenger RNAs. So the issue is how we can go. One way is to expand the population, the diversity, and another way to
Comment.
other comments. Okay, chance. Yeah. I'm afraid I'm leaving the discussion.
Topics. A. Spatial technology alters in fundamental ways single cell data from discontinuous to continuous
Okay, so that's.
because of cell segmentation and other factors. We address the problem with BRAC, entropy 2050.
That's an issue of the next discussion.
Yeah, this will come later. Cell segmentation will also be a topic, also from another breakout session on computational tools. And the other comment was,
Yes.
because of uneven technical advances, transcriptomics, not proteomics, will define initial versions of the X.
Oh,
A recent cell genomics paper on macaques
yeah.
highlights the differences and the shortcomings of a pure transcriptomic-based cell classification. We need to keep an open mind about that. Other papers on this topic are in the person shares.
Yeah, nature.
Yes, thank you very much.
Protein stuff. Jay. Yeah, comments from the transcripts.
Yeah.
No.
So I'm biased and also transparent. But I guess you asked about diversity, and I think there are multiple parameters we have to consider. One is yes, we can get many donors from Asia or Africa or South America or just underrepresented regions. But what are you going to do with that data? And as you heard from the keynote speaker, if you can get complementary whole genome sequencing data on the tissue and look at their EQTL effect based on gene expression, and that gene expression could potentially be associated with protein or RNA, or spatial association, spatial QTL can still be inferred.
So
simply the layers are different protein, the genome, and the protein. Yeah, the messenger is so.
So I guess the question is, why do we want that? Why do we need diversity using spatial? Is genetics diversity, and maybe there are visual diversity, environmental diversity. And so based on that question, we made our different approaches. Perhaps we need to do microbiome detecting bacteria together with skin or together with the gut, and that has huge impact on the environment. So I think we do have to frame certain questions to apply the right technologies. But fundamentally, even we have all these technologies that are accessible, if you're not able to afford them, you're not going to be able to generate the data. And that is a big issue, just historically, that low middle income countries are not able to afford any of these technologies that Western countries have access to. And that is why causing the most majority of this lack of diversity. Right? So there are multiple factors that we have to consider, not just scientific questions.
But maybe that's why combining RNA with H&E and then using AI to actually infer such modalities from managing alone, that could sort of bridge or
Yes, that would be the Holy Grail.
all right, just for the interest of time, we'll move to the second question, unless there's someone that really wants to comment further. All right. So next,
Can I ask a small question before going further? I'm sorry.
yes.
And my impression is RNA, technically speaking, the RNA analysis is quite mature. And we have less kind of effort on the, say, normalization or standardization of the, the genetic data set. That's one aspect of the JPA have mentioned. But for the protein analysis, I want to know I want to know the comments from the human protein atlas people. So can you say what technology is already mature for the normalization? Because my knowledge is quite old about the master especially. So and when we have had kind of big problem for standardization. I'm sorry for I'm just
I think you're asking wrong. Definitely not.
Yeah.
Very RNA focused.
Yeah.
I-
Because
I-
you started with protein.
I think that spatial proteomics was there.
No, it's
has been there.
In general, mass spec for the antibody based was
That's been there out there for long now.
quite stable.
Sorry?
Quite stable, robust.
I would say so. I mean, there are also multiple platforms and
Yeah,
technologies
I agree.
using different chemistries behind, and one can discuss exactly what would be the most interesting. There are some which are not really antibody based, etc. But I think that actually there are a couple of spatial proteomics techniques that are being widely and broadly used. Also, in particular in clinical settings. So
Okay. I see.
robustness shouldn't be an issue.
I'm sorry, Josh.
No,
Let's move on to the
I think those are important questions, so it's good that we have them and we talk about that. So, second question: should we combine atlas generation efforts with imaging, with capture, and it's all 3' or pro-based, or with both? And when deciding, we should take into account benefits and limitations of this, and in particular, wet lab and data analysis considerations. So how do we think about that, considering that we have the bio networks that will need to use these techniques, and that we have the computational people that will need to analyze the data generated with these techniques?
Yes, Jay.
So if you were to take the analogy of single cell efforts to build the initial draft of the human cell atlas, most of the cell type annotations have been used only using the protein coding gene or 3' gene. So in essence, if you were to use pro-based, it's more than enough to annotate cell. And this is a human cell atlas, not human gene atlas or human gene annotation effort. We're not here to discover new genes. We're here to discover new cell types and maybe cell states. I think just focusing on, it's just my opinion, is more than enough to annotate the cell types. With that said, yes, then you ask what are the further advantage of doing 3' for spatial? Because then, or maybe you could do a taxi based on the spatial. Then you're getting to gene regulation, non-coding RNAs, and that space. And the question is, how much of those information will enhance the cell annotation effort? And what have you learned from the single cell efforts, and how can we accelerate and translate that into spatial?
Although now, as soon as the human atlas moves also into the disease spectrum, eventually then not only cell annotation, but also genes or differentially expressed genes might have or might play a more important role.
Yes.
So we might want to have also more unbiased view on tissues as well. I'm just putting out there. I'm not.
Yeah.
Let's start. Yes.
Yeah, I fully agree with that comment. Especially if you want to kind of learn about perturbations of tissues, then really need to go with much more exploratory approach. And in that sense, also going back to the previous question, I think it would be ideal to have at least a selection of organs that we can screen with all the different type of modalities to really learn the added value of each one of them. And also that should kind of enhance building good virtual cells models, I guess. And from that, we will also learn what are the key modalities that we come to miss on. So,
So sort of a pilot covering most of can be covered, and then
this
from that.
I agree with Stefania. My comment originally was with the pro-based, although they're great because they're very sensitive, very specific, the images are great. If we all use a different panel, then how are we going to build the atlas together? So that is something to think about. Although we like to have a panel for the lung, the heart, the brain, but then we can't do the cross-body integration. So that just I'm thinking, just saying where it's coming to my mind, but.
So then we would need to agree on one or different
It's
panels.
a set of genes that everyone runs at least, and then maybe have organ specific, something like that. But if we all go everyone on their own way, then we can't reuse those data.
So, any comments? Yeah, I agree. So, and speaking of genetics, and as you mentioned, so the genome sequencing or the GPAS data is coming in respect of the same genes. Transcriptome maybe, and it's a specific thing for the transcriptome analysis. When the genetic analysis suggests some involvement of some genes, and there is no way to specify which organ is affected. So the catalog should be the base to for defining those kinds of analysis, as we presented, demonstrated by the speaker in the afternoon session. So, and to enable that kind of analysis, I agree, the more comprehensive level. So the catalog is, anyway, this is the initial efforts for us, the worldwide efforts to make a catalog of the cell, single cells. So maybe we better not to, that's my personal opinion. So maybe there is the way of thinking is maybe different from focusing on a specific genes, set of genes. Yeah, focusing on some specific diseases. And any other, maybe advice. So any other kinds of videos?
I mean, I think the larger the imaging gene panel size is, the more redundant it becomes with based methods. You would basically double the cost.
Yeah.
There are practical reasons not to do that. But obviously you can start with the more discovery, high plex assays, and then eventually work on probe sets that are like HCA, fine, tissue specific. And then I think those methods are less redundant if you can combine a sequencing-based assay, which captures just more of the transcriptome beyond coding genes as well, and then have a more cost-efficient probe design on the imaging side.
We will have a discussion on the cost, maybe later.
Yeah, and talking about probes and pre-design panels, right? So customizations, pre-designed or combination of both, and that's per tissue. Also cost efficiency. Holger just brought that up. And this also leads to the next question, but I'll put it out there because it's again about how do we design panels. Do we want to do cell typing per se? So we basically, that's our first priority. We build panels on marker genes, and how we do that across tissues? Or do we also think more on cell-to-cell interaction, communication, or pathways that are sort of more comparable maybe across organs or even across diseases? And I see there's something to say.
I'm just going to comment. So when we think of spatial experiments, we do the ground truth first. We do the
Yeah.
H&E first. We start off with the pathology annotation of the tissue. That's ground truth. That then informs us on fields of view or regions of interest, all of that. So is the framing of our starting point different with spatial? Like, should that layer in? Because fundamentally, whatever you're calling, you need to be able to establish that, that's a real cell type based on a pathology annotation of that. So I'm just thinking about that's a cheap way to identify your regions. Histology.
But how do we bring that into the panel design, let's say? How would that help us defining better panels?
In case of diseases, it's preset, a kind of in the
What do you
for the
mean?
case of the lung cancers, for example, the key genes are already set, kind of established. And maybe it may not be a bad idea to start with the already established set of the genes. And then for the pathology as well, and they're already in terms of the diseases like cancers, the lesion of interest is already set. Pathological. So maybe that's easy. That's your point, right? Just for saving the money and the J. I knew you were making a comment of video.
So you mentioned disease, so do we need panel for healthy and disease?
Yeah.
How do you define disease? And when do we know we covered up disease?
Yeah, I was thinking of the cancers because it's not
Even cancer, so like when do we draw the line? I think healthy probably may be easier, but if you start getting to disease, you may discover new set of genes that we would never.
Yeah, you're definitely right. If we are thinking about the say psychiatric diseases like ASDs, we don't have any idea what kinds of organs, tissue types are affected simply by looking at the GWAS things. So we have to almost no pathological, inspection is thing. So you're right. And the human cell atlas is of course expected to cover those, undefined diseases as well. Maybe cancer is
Getting old is disease?
yeah.
The whole story,
It's funny.
there's a growing, now you can do probes that are almost, wide.
Yeah,
Transcript.
that's right.
Yeah.
I think designing probe sets that are around like 1,000, 2,000 genes, we have to come in with a strong hypothesis that we know most of the cell types and states already, that our single cell atlas are just comprehensive enough, so we know the plasticity states of human biology. I guess it's not true for healthy, it's definitely not true for disease. So working on panels on a diseased tissue, I think is about is what we have to take into account. And then maybe a sequencing based approach at the same time might cover that lack of visibility somehow.
Yeah, I also want to hear a comment from 10x because you have just started to launch a 2K option. I kind of expected that. And it's not just in the middle, say panel and 5K and 18K eventually. So and how do you think like this fits to this human cell atlas? Yes, yeah. And just in
Yeah, I guess it seems to me like at the discovery stage, you're going to want a essentially whole transcriptome panel.
Yeah.
And I think that could be extremely powerful. Why follow up with more targeted panels if you're getting some extra performance or cost reduction or throughput increases? So,
how do you think about customization and the per organ tissue thing?
Yeah.
It's just in the middle.
I mean, I think we're super supportive of, if the HCA wants to have certain well-defined panels that are promoted and blessed and economically made. And I think we're thinking about and working on things that are going to make it easier to make things modular. So you could have like HCA base panels, and then people want to add on their disease genes, or they want to combine things, and to do that in a kind of low slide number customization way, make that affordable. But I think everything we're doing this, how do you get the most genes so you can be, universal and not have to make decisions and choices and things like that. So
I agree. 400 may be too small. Yeah, the number is too small, but 5K may not be. No. Okay, so
One is the cost and the other is the sensitivity. all the transcriptome panels are getting less sensitive. So
Breakout 2K is no less sensitive than, yeah,
so
okay.
I think that's, I mean, that's a question basically. If what people see if it, for 1K panel is more sensitive, to pick up transcript genes as compared with 5K, 6K. Or I'm not sharing that.
Who's not sure?
It's a good question.
The challenge. I think this, to some extent,
No.
I think to some extent there will always be some truth to that. At the same time, I think there are trade-offs that will increasingly go away. Like I don't think the trade-offs of today's technology is going to be the trade-offs of tomorrow's technology in the same way, right? So again, I mean, not everything can be here instantaneously, but I do think a lot of the way that people are thinking is kind of like assuming that you have to make these hard trade-offs, and I think it's our job as the technology builders to try and remove those trade-offs so you could, as much as possible, have multiple things that you want. You say you can't do all that instantaneously for you, but I mean, that's why it's helpful to hear these things.
Including reduced cost.
Yeah, including reduced costs. Like I try to make that point to talk. I mean, I think we've, not that we're done, I think we have to learn on single cell. I know on spatial that hasn't come yet, but that is like very much something that we are really dedicated to. It's not like not just saying that, like we are aiming to substantially reduce costs for everyone.
So
Yeah, yes, that has already been touched upon slightly. So we definitely need cell typing markers, but maybe we don't need 20 per cell type. We can leverage the human cell atlas efforts that have been done already to learn what we need, and then in a modular way, maybe add other markers, disease specific or organ specific. Do we need to generate matched single cell data?
any comments? No. So then we start. Then
I would have
yes,
loved it.
yes, please.
I think it's tempting to do that because we still assume single cells is better data because we don't have the cell segmentation. But it's doubling the cost. But still for many projects we do it for the reason we're assuming it's better data that is then transferable, which is also assumption that it's not really true most often. But yeah.
I think so far, at least on my experience, is that for some technologies still helps to annotate cell types in space a lot. It might not be true for all of them, but it is true for some of them. It's not yet a solved problem how to define marker genes out of spatial transcriptomics data, and having matched single cell data set helps. Now there are everyday new data sets available, so we don't need to generate new matched single cell data set from every spatial experiment. Definitely doubles the cost.
So I have a related question to Jay. So from the human cell atlas, to what extent do we expect the single cell has been covered already? So that's depending on the coverage of the single cell sequencing data. If it's almost entirely covered from the normal tissues, maybe this may not be an issue.
Well, but as Abhi mentioned, this morning, there are 18 biological networks and attempts to complete these atlas for individual organs. And so you probably need to talk to individual network coordinators. But they're releasing the Atlas 1.0, which means there will be 1.5 and 2. So it's a growing, continuing effort, which I think is probably not stopping the foreseeable. But I think at the same time, again, I think this question is very important.
Yeah,
exactly. How do you transition, or do we need to transition?
Okay.
So a couple of comments. For some atlases, we mostly have single NUC data. And so although we think they're very informative and we're very happy with it, we're still missing what's in the cytoplasm. So I think we're
That's
not
what should be
done.
complemented by the Spatial.
Yeah.
Okay.
And then in terms of going back a little bit to what you were saying before, do we need a single cell match data and linking to this is, do we have an idea of how many cells we need to find the very rare cells? And then every time we introduce a new disease or a new group of patients, there might be unknown things. So I think we're very far from being done.
You're right.
I mean, from what we've done so far, when generating matched data sets, spatial and single cell, and then trying to integrate, actually, your question is very relevant because we were generating single cell data on 5,000 cells, and we were actually annotating 5 million cells out of these 5,000. So then we also had the question, does it really make sense at all, no?
So another choice is we have just heard the scaling a single cell to this 500,000 of cells is in it.
Yeah.
So that's really becoming issue. Okay, so I don't know.
Okay, so and 3D, how do we get 3D data?
Oh yeah.
Do we use different Z sections as it was possible until now, or we need to implement in the human cell atlas technologies that allow us to generate 3D?
Still a lot of things.
Yeah, maybe I'm just adding an extra question. So is more the 3D level those technology can achieve? Is really the 3D level that we envision for an atlas?
Good question.
Have you even tried, for example, ceramics? Because this is my first time here.
I think they are the sponsor. So.
Yeah, sponsor, yes. That's the reason why I know that technology. So, yeah, this.
I can add another question, because in the end, with the, let's say with the cellular mix and the 100 micron, you will have a full cell and neighboring cells, but you will not have a 3D in organ section.
No.
It is just, I think it's more comprehensive on the spatial cellular side and spatial cell interaction, but it's definitely not what we're aiming at, reconstructing a 3D organ or tissue. That's not an answer, it's just another challenge to solve.
Probably maybe the answer or the solution for both of the questions is to again use AI tools that will allow us to sort of reconstruct everything that is not being literally analyzed. I assume that if you already start from a fully 3D 100 micron section, that gives you a better starting one.
That's the issue of discussion. And so the human brain atlas, Alan Atlas, has generated the data for the entire mouse brain and the 3D matter. But I felt that it's been a really laborious work, and there are lots of technical difficulties for here. So, and I'm quite, doubtful whether we can do the same for the human organs, which are usually much, thicker and a bit bigger. So, but yeah, I agree. That's worth doing to understand the real relevance of the single cell diversity that we have organ. So, yeah, any opinions? And yeah, it's.
So I think the technology will evolve and advance. They'll be cheaper, but key is to hopefully all the people in this group start annotating.
Yeah.
Creating very well documented metadata for your tissues that you're acquiring. And if you're able to provide this common coordinated frameworks where you can actually annotate which anatomical region of the organ tissues are coming from. At least that could help later on actually reconstruct. But yeah, I think just for the group here, can start annotating these issues well.
So that's a bit on the direction on what Utama was saying, that what from where you start from for spatial experiments, basically the HME and a good pathological annotation.
Yes.
I just wanted to point out, the mouse brain atlas is not volumetric. It's still sampling. There's
So,
like
okay,
200
okay, I see.
sections within seconds. So, yeah, I think whether or not you need volumetric 3D information depends on the question and orbit.
Yeah, that's good.
So, that thin section, adequate sampling gives you the coverage of the cellular distributions quite well across most organs. But sometimes you need that extra information. So in the brain, for example, if you want to understand cellular organization as a whole, that's enough. If you want to understand circuit organization, then you have to go volumetric. You can't lose information across longer distances. So I guess I would ask this too, what you need for your particular organ may not be a one-sided.
And I think that's also depends on the research purposes. For example, in case of cancers, and even though the cancers are separated into two places on that XY plotter, we cannot 100% sure those cancers are really separated. If there is a round blood vessel connecting those two places, it's not made right. So, have you ever seen, let's say, conjunctive sections in terms of the spatial distribution of the cancer cells, for example? Maybe I ask you other questions again.
Yeah, I mean, you're moving through that volumetric space. You don't know if antibodies penetrate that deep. There are challenges in doing that. If you do 500 serial sections, you're going to annotate each section, right? We work with pathologists that look at 5 micron sections. And so we take the next serial section and annotate that. Peter Soga will tell you need 35 micron to be able to see that full cell. So I don't know how deep you need to go, but also, do you lean on numbers by patients or disease, or going one sample really deep, right? So these are I think they're challenges for the field that everyone's sort of thinking about, but there's no clear path.
Yeah. I'm sorry, we're quite well behind the schedule.
No, I think we're actually at the last question.
Wait,
Maybe you should just quick. Since I'm still a representative in the room, maybe I should say something. I want to echo what Ed said. It really does depend. It seems to me, and one of the questions that you're asking, at Stellarmix, that we're going to be expanding much beyond just the narrow Z direction that in XY that we're doing, and you're going to see the samples get larger and larger. But the truth is, you may not need 3D for everything. But I will point out that at least in our early founders papers, they showed that 2D methods definitely miscalculated cell-cell interactions. And in the cases where this is important for the human cell atlas to know, it seems to me that it would make sense to use 100 micron sections as your starting point. You still have the section. I mean, we're not doing the whole brain, we know that, but instead of doing 10 microns, to reduce your labor in your process by 10-fold, looking at 100 microns seems like it might make sense, but maybe not for everything. And I'm actually here to learn from you. I'm a little bit surprised that there's not more of an interest in trying to reduce that labor. At least I'm hearing this one, but I think in certain questions, it will help get accuracy up. And so it really depends on how accurate does this human cell atlas need to be, I guess.
Jay, any comment? From the general assembly, from the HCA. The goal of the HCA.
We're at 250 bucks today. And I know that's too low, but it's going to change. The original paper showed 5,000 bucks. We know that this will scale. It's just a matter of making that work every time their product, and so we're doing this.
Can I use the chance to ask you a question that I didn't have the chance to ask you before? How would you actually define the cell boundaries?
Yeah, great question. It's the first question I get every time I present. And right now, we're doing very simple DAPI staining and amplicon clouds, right? And we're extrapolating from those two pieces of information. However, we have an active program in cell segmentation right now, and we're making actually really nice progress. So I hope to have some more updated information for you, but this is hugely important. And we have, being a volumetric and a 3D method, we have a unique ability to really map those cell boundaries in a way that maybe is not possible in 2D methods. So if you have an opportunity here, we know we want to make sure that we execute on it.
Is there any other comments? Yeah, thank you, okay, by the way. But,
You guys can.
Yeah, thank you.
One more time.
Yeah.
I agree with the last comment. Yeah, I think 2D is always a very poor proxy of how cells interact. And because there are many cells in the surroundings, I just don't see. You assume that interaction between neighbor cells is influencing of interest. That's probably not true, and that's true if you look at the other neighbors. But I think the reality is that technology or 3D technology is where 2D technology has been 5 years ago, or single cell had been 10 years ago. So it's I think foreseeable future that technology is mature. There would be hopefully just one company, but maybe many others that would allow us kind of to build our business more holistically. But I mean, it also have to be realistic where we are today and what we can do today at scale.
Any other comments? Only looking. This is
This was our last question.
Okay, that's good. Sure. Maybe we can go on in general, so we can cover all of the issues for the discussion, and especially I would like to encourage that those who have not spoken anything during the discussion, please raise your hands and say anything, and your comments will be fed back to the General Assembly. So it's a rare chance we can make feedback. Yes, yeah. So any comments from any parts of the world?
Okay, we have another comment. Keep in mind that at cell level, pathologists are quantitatively near blind. I hope that's no pathology. Not much reliable. Then has citation and seeing or behaving in hyperplex spatial proteomics by antibodies, no biases for an image-based technology. I think there's a lot of quantitative biology already, and in anybody, it's true that this anyhow probably was related to a previous question. Any other comments to close the session? So thank you very much. There's one. Okay. Yeah.
Thank you for comment.
Yeah, just more of a question, putting a question out there. I understand now spatial technologies have their own limitations. Single cell had their own. So comparatively, putting it out there, for example, 2D space versus what amplification did or dissociation did to single cell. Do you think that both put together will fill this gap? Or there has to be something else that comes in the picture which kind of completes this picture?
Yeah, any comments? Again, yeah.
No, can you? I didn't really get the question. Sorry.
So from single cell technologies, there have been limitations on the amplification side versus also, in addition to that, what dissociation does to cells. So do you think the current spatial technologies will fill that gap, or there is more to be done on those sides?
I think at least partly, there are some of the problems that are being covered by Spatial, right? So bias on cell types by generating cell suspensions. You get everything now from a tissue as far as your panel covers for it. If it's imaging based, if it's sequencing based, you can definitely map any cell type on any organ. So that's covered. We really get a better real picture of the tissue population in terms of cell types. What else?
That's maybe so that's maybe our previous discussion about to what extent we want to go into details. And for the single cell sequencing itself, we can stop at the very narrow sequencing there are coverages like that. And we can go in further in terms of sequencing there. And if we want to keep the spatial information, of course we can go to the spatial. That is complementary to the single cell analysis. But when we come to the spatial, the sensitivity may be different depending on the platforms. And there's some of the delegates on the internet mentioned that the cell type separation may not be always when we're simply focusing on the messenger RNAs, even though it may go into quite deep into the transcriptome. So that is perhaps the filling the gap is having a different needs. So of course the more the better. And if we put more efforts, then lots of focusing let's say they're employed by the various kinds of platforms. Of course we can expect greater less gaps. But it's the more costly and technically difficult. So it's a trade between the precision and the cost also. Maybe. So any other
Probably we should go to the auditorium, right? Okay, so thank you very much.
Thank you for clearing that comment. I'm sorry, I'm afraid I'm not
Okay, that's a good session.